March 22, this is the last exam that happened in India last March. Okay, nothing new, same. Question number 1. A student investigated the effect of surface area on the rate of diffusion. Since he's telling you he investigated, what did I want to see the effect of? I wanted to see the effect of surface area. So I will change the surface area and check what will happen to the rate of diffusion. So I underlined the surface area, and this is the factor changed. The student prepared different size blocks of agar. How did I change the surface area? I got different sizes of agar blocks. The blocks of the agar had different surface areas. I hope this is the blocks. Each of them have different surface area, total surface area. Okay, the agar contained universal indicator, which made the agar green in color. Since all of them are green in color, this means that all of them have the same type of universal indicator. This is a controlled variable. What's a controlled variable? It is something you did the same in all the blocks, the type of universal indicator. I underlined it and I wrote controlled variable. The blocks of the agar were put in hydrochloric acid. The universal indicator turns red in the presence of acid. What will happen in this experiment? If you put the blocks in the acid will diffuse from high concentration to low concentration. So it will change the green color to red. Type. Let us see the steps. As the acid diffused into the agar block, the indicator changed from green to red. What am I going to measure here? What are the results you are going to measure? You are going to measure the time taken for the acid to diffuse to the center of the block. Step number 1, 10 cubic centimeter of 1 mole per cubic decimeter hydrochloric acid was put into each of the 4 test tubes. Since you put the same volume of hydrochloric acid in the 4 test tubes, also the same concentration of hydrochloric acid in the 4 test tubes, then these I will call controlled variables. What else? The students used a ruler and a knife. There is a knife, then this is a hazard. This is something dangerous. If I should take care of, probably he will ask about it later. He used the knife to cut the 4 blocks of agar, A, B, C, and D, from a larger piece. Each block had a different size like this. Okay. Step number 3. The agar block A with this size is placed into one of the test tubes containing hydrochloric acid. A stop clock was started. The color of the agar was observed. What's the problem here? Again, the color of the agar block was observed. This is considered to be an error because it will be difficult to determine the end point. You won't be able to determine when exactly the color change happened. Okay, step 3 was repeated with the remaining blocks B, C, and D. He did the same thing with the other 3 blocks. Of course, he will ask you to draw a table. Figure 1.2 shows the time taken for each block to turn completely red. Prepare a table, convert the times into seconds, and record them in your table. This is a very easy table. We know previously, what do we put on the left side of the table? Whatever you change, you put it on the left side. The results, you put it on the right side. So what did I change in this experiment? I can write multiple things. But in this test, he considered because he didn't give you the exact values of the surface area or the values of the volume. So if you wrote on the left side the name of the blocks, block A, B, C, and D, correct? If you wrote the surface area and you calculated the surface area, the total surface area for all the blocks, it's correct. If you wrote the volume, برضو correct. طيب, this is the table. Here I wrote the block A, B, C, D on the left side. These are the changed blocks. I wrote the volume. I calculated the volume for each block. يعني 1 times 1 times 1, 1 times 1 times 0.5, 1 times 0.5 times 0, side times side times side. This is the rule for the volume. Or I can write the surface area, which is يعني makes more sense. It's better to write the surface area, but all of them are considered correct. How do I calculate the surface area for the cube? It is side times 6, because you have 6 surfaces. Okay? And then I write the results. Time taken to change the color. Here are the times. You have to convert it to seconds. How will I convert it to seconds? 6 times 60 plus 56. 3 times 60 plus 2. 1 times 60 plus 54. And I write the numbers in seconds here. Please don't forget to put the units of the headings you did. If you only write one column, يعني if you write volume without the block, it is right. If you write block without the volume, it is right. After the table, he asked you, state a conclusion for the results of this investigation. The conclusion here, as the volume decreases, the time taken to change in color also decrease, okay? Or if you talked about the surface area, it would also be the same. Yani if you wrote here the surface area, you will have the number. So you can say that مثلا time taken increases as the surface area increases. It will also be the same. Yani the conclusion according to your table. What did you write in your table? Write the conclusion. State the variable that was measured. What is the results you measured? Easy question. The time taken for the agar block to change in color. This is the results you measured, which is the dependent variable. But what is the variable changed? What did you change in this experiment? We changed the surface area. We decided this, or خلاص we underlined this from the beginning here. We changed the surface area. طيب. What are the variables that we kept constant? Anything from the experiment. There is a lot of things he said, like the type of indicator, like the volume of hydrochloric acid, the concentration of hydrochloric acid, or even if you said the type of acid in the tubes, I put all the hydrochloric acid in all the tubes, كل دول, all of them are considered controlled variables. Okay, suggest one way of improving the method used in this investigation. As a general rule, improvement is always repeat the investigation. Repeat the investigation is always an improvement. طيب, what else? Control the temperature. He did not say anything about the temperature in the experiment, and this is an experiment on diffusion. We know that if the temperature changes, it will also affect diffusion, so I can say control the temperature. طيب, this is something to take care of. We said in this experiment that it is difficult to determine the end point, or in any experiment that the color changes, and you would not be able to know when exactly did the block turn red. So what is the improvement? What is the solution for this? He wrote it back in this exam. Use video to observe the color change. You will record and then you can watch the video. So it's easy to repeat and determine exactly when the color change happened. Okay, describe how step 2 could be carried out safely. Step 2, what was it? Step 2 was cutting with a knife. And we said that this is a hazard. So how could it be done safely? Cut on a cutting board, cut on a flat surface, cut away from the body. All of these are correct. Okay, the table shows the surface area and the volume of the blocks that were used. He calculated the surface area and the volume. And then he wants the ratio of surface area to volume. Yani it's very easy. You will just calculate the ratio. It will be 2.50 over 0.250. It will give you 10. This means that the ratio here is 10 to 1. So the ratio will be 10 to 1. C, many organisms have adaptations that increase the area of gas exchange surfaces. This is a photomicrograph of part of a fish gill. Okay, this was a bit of a tricky drawing, but it's the same rules. Draw a large diagram of part of the fish gill. What should I take care of? There is هنا two كده semicircles, and this semicircle, I should draw a double line to show its thickness. And then I have branches on both sides, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, okay? So I drew it like this. What are my rules? Large drawing, no broken lines, no broken lines. What do I mean by broken lines? The lines should be smooth. Don't do this and then do this and do this. I don't want to see these connections. If you did it, erase it and connect it again. No shading. Do not color anything. I want it all white. Okay, just outlines. And then the branches like I have here. Okay. But after the drawing, he will give you the magnification question. Measure the length of line PQ. PQ فين? There is PQ. You will measure it using your ruler. And he asked you that he wanted in millimeters. When you measure in centimeter, and then you do it times 10 to convert it to millimeter. Calculate the actual length of the fish gill. How do I calculate the actual length? I wrote my triangle, magnification equals image over actual. I want the actual, so it will be image over magnification. This is my image, and here is my magnification. Divided, it is 0.1636. Take care of the roundation. Here he asked you to round it to 3 decimal places. So this is 1, 2, and 3, 6, round up, it will be 0.164 millimeters. The last question, not the last question. Fish is a source of protein in the diet. How could you test to show if it contains protein? This is the normal food test. Please, please, please study the food tests, very important. Add bioretry agent, the color changes to purple.
Okay.
Okay. The last question in question 1 is, the photographic film consists of a plastic sheet coated in crystals. Photographic film, this was used in the all-day cameras. The crystals are fixed to a plastic sheet by gelatin, which is made of protein. If the gelatin is digested by protease enzymes, the crystals fall off and the film will become transparent. Please, please, please don't panic when you read something like this. If you do not know what is the film, what is the crystals, doesn't matter. At the end, he is asking you about an enzyme. Okay, protease enzyme. Plan an investigation to determine the effect of temperature on the activity of protease enzyme. Very basic experiment. Okay, we know it. It's nothing. The only thing that you need here is what results will you measure? He gave you a hint. If the gelatin is digested by protease, the crystals fall off and the film will become transparent. For this experiment, we will follow the same 7 steps. Number 1, use 5 films. Number 2, what will you put the same? Same type of film, same volume of protease enzyme, same concentration of protease enzyme, same pH buffer solution. You have to put them in the same pH because you are changing the temperature. Step number 3, what are you going to change? Put each in a different temperature. And I gave him example. This is 10, 20, 30, 40, 50. Step number 4, what are you going to measure? Measure the time taken for the film to go transparent. This is the hint he gave you. He told you that when the protease works, it turns the film to transparent. I am going to measure the time taken for the film to go transparent. Type, what is an improvement I could make? Since you have temperatures, then I should tell him, maintain the temperature using thermostatically controlled water bath. Safety precaution, wear goggles, repeat at least 3 times. Just like any other experiment we did before. The first question is 27 marks out of the 40. So the rest, the 13 marks, is for question 2. It will be the graph. The cabbage leaves contain vitamin C. Boiling the cabbage in water reduces the vitamin C content of the leaves. Some students investigated the effect of boiling time on the concentration of vitamin C. يبقى he will boil the cabbage and he will check the concentration of vitamin C. The student prepared the cabbage samples using this method. Heat a large volume of water until it begins to boil. Measure the mass of chopped cabbage. Place the chopped cabbage into the boiling water. Continue to boil the water. Remove a sample from the water at each time interval. The first question he asked you, state 2 pieces of apparatus other than the safety equipment that is required to carry out the steps. Other than the safety, يعني don't tell him safety goggles, don't tell him gloves, don't tell him lab coat. I want equipment I need to do this experiment. Easy. You want to measure the mass, so you need a balance. Sorry, this is a water bath, not thermometer. You want to heat the water until it boils. How are you going to heat it? You need a water bath or a beaker and a Bunsen burner, okay, to heat the water. These are the equipments you need. Okay, what else? The vitamin C concentration of each cabbage was then determined. He gave you the results, but these are the boiling time, and this is the vitamin C concentration. How am I going to draw the graph? Plot a line graph on, of the data. Left side of the table on the x-axis, right side on the y-axis. First thing to do when you get the graph, please put the labels. You will take 1 mark if you just put the labels. Boiling time / minutes, and here is the vitamin C concentration / milligram per 100 gram, all the unit. Number 2, put the scale 0, 5, 10, 15, 25. Do I put the numbers like they are? No. Why? Because here I have a difference of 5, difference of 5, difference of 5, difference of 10. So I can't just put the numbers like this. I have to have equal difference. How did I put it? 0, 5, 10, 15, 20, 25. What about the y-axis? I have from 13, this is the smallest number, 34, this is the biggest number. So I can say that I need my scale to cover from 10 to 35. Do I need the numbers before 10? I don't need them. I don't have points before 10. This is why I started from 10 and I went with an interval of 5. 10, 15, 20, 25, 30, 35. Next, you plot your points. Please, please, please. The points is either X or a dot and a circle. I don't want to see this. You will be penalized for this. You will lose marks if you did this. After you plot your points, join point to point using a ruler. Since it is a normal line graph, you join point to point using a ruler. كده, you will take the 4 marks for the graph. Use your graph to estimate the concentration of vitamin C in the sample of cabbage that has been boiled for 20 minutes. 20 minutes, he wants the vitamin C at 20 minutes. So how did I do it? I have to draw the line. Draw the line from 20 minutes up to your graph till you meet your graph and then go left, okay? You have to show on your graph how you did it. And then you read the number from here. For me, it's 17.5, so I put 17.5. Any number is correct as long as you read it properly from your graph. Okay, let's do the last page and then I'll take your question, ya Hamza. Table 2.1 shows that the start of the investigation of the unboiled cabbage leaves contained 34 milligrams of vitamin C. At the start, the unboiled cabbage had 34 milligrams of vitamin C. Using the information in the table, calculate the percentage decrease in the vitamin C content after boiling for 25 minutes. But let's go back to the table. After 25 minutes, how much was the vitamin C? It was 13. At the beginning, how much was it? It was 34. How do I calculate the percentage decrease? The normal rule, final minus original over original times 100. Or you get the change between the numbers, you get the change between the number over the original number times 100. 34 minus 13 over 34 times 100. It gave me 61.76. Take care of the roundation. He wants it to 2 significant figures. So these are the 2 significant figures. So the result is 62. Okay, the last question. The student wanted to know if the vitamin C had moved from the cabbage leaves into the water that the leaves were boiled in. How could he test the water for the presence of vitamin C? This is the test for vitamin C. Add DCPIP, which is the reagent test for vitamin C. The color will change from blue to colorless. And this is the March exam.